Welcome back again with a very interesting one-of-a-time topic, prokaryotes. We know these cells that have roamed into our lives without even taking our permission, these structures that have actually succeeded in their functions. Now let's know more about them. They possess a circular DNA that's not enclosed inside a nucleus. As you can see here in the diagram, it lies freely in the cytoplasm. It's not even coiled around histone proteins. Imagine with me, it's like something you coil around, like a tube, something like that is coiled around. It's supposedly coiled, but now it's not. It's circular. It cannot react. It's just closed everywhere, which means it cannot also be involved in either mitosis or meiosis, cell division structures. All of that is not present in their life, okay? It's not in their cell cycle. It also doesn't possess any membrane-bound organelles. Of course, we know mitochondria, chloroplasts, amyloplasts, and all these structures that are actually present in the eukaryotes. The mitochondria that gives the cell its energy. They can also actually have their energy, but it's not of the same topic that we're talking about now. Okay? This is the mitochondria. It also has a special type of ribosomes. As we've said before, the ribosome is made of the ribosomal RNA and polypeptides proteins. Okay, that's what the ribosomes are formed of. It actually contains two subunits, and this one is very different. The 70S ribosome is a smaller type of ribosomes. It actually exists as 18 nanometers, which is like the smallest thing you'd ever seen in your life. You couldn't even see with your eyes. It can only be seen by an electron microscope. Okay. It also has these structures which might confuse you just from their names. Pili, which are these structures coming out of them. We're going to know more about their function, but later on the next slide. The mesosomes, which is an unfolding of the inner membrane. It's sometimes said by scientists that it's for photosynthetic membrane uses, but it's still not discovered as the other structures we're going to discuss now. Also the plasmids, which really interests me. These plasmids actually carry some genetic material that makes this kind of prokaryote able to be like non-existent to the antibiotics. Whatever you take, I'm not gonna die. That's what it says, okay? It's resistant to these types of antibiotics, okay? It also has a peptidoglycan cell wall, aka or also known as murine, okay? It possesses a cell surface membrane, of course, which is actually more permeable than the cell wall. You know, the cell wall prevents the cell from bursting and gives its turgor pressure, but also the cell surface membrane controls everything. It can let a structure enter or not enter. It's more like controlling the permeability, okay? We also note that the mesosomes are an unfolding of the inner membrane. We can also draw a 2D shape together. It is said to be a photosynthetic membrane, yes. The loop of DNA is naked. By that I mean it's not coiled around the histone proteins. Yeah, and just as we said, they cannot undergo mitosis and meiosis because of the absence of a nucleus. The nucleus is like the major thing going on in any cell division. Like you see the pictures of the chromosomes moving in its phases, but not like the cytoplasm, not like the other things going on which are focused at. Okay, it doesn't have a nucleus, which is like a huge, severe problem. Okay, it has circular loop, which is like, you can call it unreactive loop of DNA. It's not involved in these. It's not coiled around histone proteins. Yeah, that's what we've discussed lately. The 70S ribosomes are found in mitochondria and prokaryotic cells. Okay, while, you know, there's another type of ribosomes which are like the 80S ribosomes here. These, which you can find lying freely in the cytoplasm or on the rough endoplasmic reticulum. And of course, we know their like irreplaceable function. These are used for protein synthesis. They are actually the site of translation, of which you can get the enzymes like hemoglobin, fibrinogen. All these protein-based stuff are made of, or not made of, they're made by the ribosomes. These very small structures you can't see do all these huge things, okay? And yeah, we've discussed before the fact that the small ribosome of 18 nanometers, while the ATS ribosomes are about 25 nanometers. Also, flagellum, it's like a tail. You know this long thing going on here? It's like its tail. It beats for motility or movement. The pili enables bacteria to attach to the host cells. You know when you're coughing, sneezing, and dying of a specific bacteria? That's what enabled it to stick into your body cells. Okay, that's what your immunity craved you to do this, okay? Plasmids contain genes that make the cell resistant to specific types of antibiotics. It's resistant to it. It's never gonna die. It's never gonna be affected by this type of antibiotic. That's why we always prescribe, like, you should take different types of antibiotics. You should take care, or they're not for the usual or daily usage, okay? Also, the slime capsule prevents dehydration of the cell. In dry conditions, it helps the bacteria survive, which is, I might say, not good for humans. Like, we want these bacteria to die and stop devouring our bodies, okay? But now, yet, the slime capsule helps them to survive the dry conditions and also protects them from the phagocytes, or these phagosomes that fuse with it in order to digest it by endocytosis. Yeah, this can happen just because of the slime capsule. It can happen just a few cases. Now, I'd like to draw like a 2D shape for you. If it ever happened to ask, like, draw the prokaryotic cell, you're gonna be always ready for that. Here is quite a blank page in order to draw it together. Firstly, you draw like a rod shape. Imagine you're drawing a mitochondria, which is like that. Okay? Imagine this is the cell wall. When I'm drawing the cell membrane, I have to do some folding here. Just like the rough endoplasmic reticulum. See that? And keep going. I want it to be yet thin, a little bit thin, okay? Here is it. And of course, let's not forget the slime capsule, like the huge protector and of the biggest fans of bacteria. See how we go like that? It's like curved a little bit. Let us draw it like that. Yeah, that's not attached. Okay. The pili are actually attached to the cell membrane, so if you're drawing it, please don't draw them from the cell wall or the cell membrane. So you better get them out of this. Okay, you can add like a structure, it's okay. Or even if you leave it like that, like totally fine. These are 3 pili. Of course it has loads of pili. And then we have the plasmids. They're quite bigger than the ribosomes, so drawing it randomly like that, roaming everywhere in this cell. Yeah, it's okay. There are 70S ribosomes. And of course, we cannot forget the loop of DNA. So let me free a space for it. You draw like this coiled electrical stuff, and then you close it. It's circular, okay? It's a circular loop of DNA. Like that, it cannot react, okay? That's what we say it. So this was like the structure we're taking. Now let's label them. This is the pili, slime capsule, cell wall, cell surface membrane, the plasmids, the circular loop of DNA, mesosomes enfolding. What else do we have in here? The 70S ribosomes. And here you go. This was the labeling of the prokaryotic cell. And that's basically what we took for today. I hope you guys enjoyed it. Thank you for watching.